In Chapter 23, we talked about how acetyl-CoA formed from each of our major biomolecules is metabolized for energy in the citric acid cycle and the electron transport chain. In this chapter, we talk about how carbs enter the body, get converted to acetyl-CoA, are used for anaerobic energy, and are stored for later. We'll also talk about the role of hormones on directing metabolic pathways. We see the same steps we had talked about in Chapter 23, now laid out specifically to carbohydrates. Carbohydrates are digested all the way down to monosaccharides, which can then be converted to acetyl-CoA through glycolysis. Glycolysis and other pathways involving glucose will be the subject of the majority of this chapter, but first, how do we get everything into the monosaccharide form? When we have monosaccharides, it doesn't take much to digest them. On the other hand, complex carbohydrates need to be broken down to monosaccharides. This process starts in our mouth, where the enzyme alpha amylase breaks it down. Alpha amylase, as an enzyme that is optimized for the more neutral pH of saliva, denatures in the acid of the stomach, but is re-released in the small intestine to further break down the remaining polysaccharide chains. Any disaccharides that remain at the surface of the intestinal cells are broken down by specific hydrolases: lactase for lactose, maltase for maltose, and sucrase for sucrose. Only monosaccharides are taken into the cells and then the bloodstream, and mainly glucose, but also fructose and galactose. This diagram shows the path we just described, some digestion in the mouth by alpha amylase, and continued later in the intestines. The intestines are filled with villi, tiny little fingers which give a much larger surface area for the absorption of nutrients. Once we have those monosaccharides in our bloodstream, they can be absorbed into any cells and used in a variety of ways. One of the main ways these monosaccharides are processed is by conversion to acetyl-CoA to be used in our central metabolism. So that's where we're heading. Most of the monosaccharides come in the form of glucose, which is broken down through a series of steps called glycolysis. Glycolysis has two phases: a stage with all 6 carbons of glucose present, and a second stage where the 6-carbon molecule is broken down into 2 3-carbon pieces. The first stage is shown here, where 2 ATP are invested. Notice this step is costing us energy for a process that we ultimately want to produce energy. The phosphates installed in this phase will be made higher energy in the 3-carbon phase and recovered as ATP. In the 3-carbon phase, we can see the actual production of energy. Notice that everything is produced in sets of 2 here. That's because we now have 2 3-carbon molecules that are being processed for each glucose that came in. We get 2 NADH and 4 ATP in the 3-carbon phase, all in the cytosol. But don't forget that we spent 2 ATP in the 6-carbon phase, so that our net yield is actually just 2 ATP and 2 NADH in the cytosol. This cytosol piece is important because NADH in the cytosol can't be oxidized in the electron transport chain directly. Let's put a pin in that and talk glycolysis first. The first step of glycolysis invests in ATP. That ATP can be gained later, but it also functions as a mean to trap glucose inside of a cell. After the glucose is trapped with a phosphate, we isomerize the glucose to a fructose, just moving the ketone over. It doesn't give us any energy, and we don't spend any energy, but it does posture us for gaining more energy later by breaking the molecule down. We can lock us into the phosphate form by adding another phosphate on the other side of the 6-carbon molecule, which helps us later. For now, it costs us an ATP for a total of 2 ATP spent during the 6-carbon stage. As we move into the 3-carbon phase, we don't split the molecule evenly. We need both pieces to be glyceraldehyde 3-phosphate, so the piece that is not glyceraldehyde 3-phosphate needs to be isomerized. You've seen both glyceraldehyde and dihydroxyacetone before. They're the simplest trioses, the 2 that you've memorized. In the 3-carbon phase, we start with glyceraldehyde 3-phosphate. We have 2 glyceraldehyde 3-phosphates for every glucose, one from glyceraldehyde phosphate produced directly and another isomerized from dihydroxyacetone phosphate. With this glyceraldehyde 3-phosphate, we can oxidize and gain energy. The oxidation stores energy in NADH, but also in a new phosphoester bond that can be hydrolyzed later for energy. When we phosphorylate ADP into ATP later, we'll gain that energy back. The NADH is in the cytosol, not in the mitochondria, so we can't think of it in exactly the same way that we did in chapter 23. We'll talk more about that later. Notice that since we have 2 glyceraldehyde 3-phosphates, we get 2 NADHs. Everything is doubled from this point forward. The phosphate that we trapped in the last step is hydrolyzed to make ATP. Again, this step happens twice since we have 2 3-carbon molecules. We get 2 ATP total at this step, but that just cancels out the 2 ATP we spent earlier. We still have the 2 NADH in the cytosol, but really that's the only thing that's our net production at this point. Step 8 is really just an isomerization, but it makes it much easier to get energy out of the phosphate that remains later. Step 9 is a dehydration. Again, this doesn't give us any energy directly, but it does make the phosphate much more reactive and easier to put on to an ADP in order to make ATP. In this final step, we finally see the phosphate removed to generate ATP. Again, since we have 2 3-carbon molecules per glucose, we produce 2 ATP per glucose at this step. This brings us to a total of 2 ATP produced per glycolysis, 2 NADH produced by glycolysis in the cytosol, and 2 pyruvates. So our net yield is 2 ATP, 2 NADH in the cytosol, and 2 pyruvate. This slide does 2 things. It shows you on the bottom left the overall reaction of glycolysis and how, as a net reaction, glycolysis takes 2 NAD+ and reduces them to 2 NADH, 2 ADP and converts them to 2 ATP, and takes a glucose and converts it to 2 pyruvates. It also shows us that while glucose is the major monosaccharide processed for energy through this pathway, galactose and fructose can also be processed through many of the same enzymes for the same net amount of energy. They just enter the glycolysis pathway at different points, but still spend and gain the same amount of energy. Now that we've talked about the entire process of glycolysis, let's talk about normal energy yields for glycolysis. Under normal aerobic conditions, that's conditions with oxygen, we'd want all the reduced cofactors like NADH and FADH2 to be oxidized in the electron transport chain to give us energy. If that was the case, glycolysis would yield 2 ATP and 2 NADHs that then we could use to make 2 times 2.5 ATP. Remembering that 2.5 ATP corresponds to the yield in ATP of NADH in the mitochondria. 2 ATP from glycolysis directly plus those 5 ATP that we could get if it was directly in the electron transport chain would normally yield a total of 7 ATP. The problem is we can't actually get the NADH into the mitochondria. To compensate, our bodies use dihydroxyacetone phosphate, which is much smaller and can move in and out of the outer mitochondrial membrane to accept electrons from NADH. The reduced form, glycerol 3-phosphate, can also move freely through the membrane and move electrons to a redox cofactor inside. The problem is that some energy is lost in this process, and now the electrons can't remake NADH, they only have enough energy to make FADH2. So the 2 NADH produced in the cytosol by glycolysis are really only worth 2 FADH2 once they're moved inside the mitochondria. Our total yield then becomes 2 ATP directly from glycolysis plus 3 ATP from the NADH in the cytosol once their electrons are transferred to FADH2 in the mitochondria for a total of 5 ATP from glycolysis, and then 2 pyruvate as well. The pyruvates that we produced in glycolysis can actually be further oxidized to produce even more energy. They can be oxidized to acetyl-CoA and then processed in the citric acid cycle in the electron transport chain. That's actually the main way we can get energy out of glycolysis. But we need oxygen to do this, so this is only the pathway when we are in aerobic conditions. That's conditions with oxygen. When we're in anaerobic conditions, conditions without oxygen, we can't use the electron transport chain. This leads to a significant problem. The NADH that we'd like to use for energy can't be converted back to NAD+ to keep going through glycolysis. So in anaerobic conditions, again, conditions without oxygen, we'll ferment the pyruvate, reducing it with NADH to restore the NAD+. This is less desirable by far. We can't get energy from the pyruvate or the NADH, but it's necessary because it's the only way to regain NAD+. The purpose of fermentation is to regain that NAD+ and allow us to get a small amount of energy from glycolysis. Anaerobic pathways allow us to get at least a little bit. Let's hone in on the differences between these fates of pyruvates a little bit more closely. The optimal route for pyruvate occurs in aerobic conditions. Pyruvate can be oxidized in the mitochondria to acetyl-CoA. The acetyl-CoA can then be used to make much more energy using the citric acid cycle and the electron transport chain. Each glucose will give us 2 pyruvates, so there also would be 2 NADHs produced by their oxidation to acetyl-CoA, and 2 acetyl-CoAs that can be metabolized in the citric acid cycle and the electron transport chain. The total yield for glucose being produced aerobically then is 5 ATP from glycolysis plus 2 times 2.5 ATP for the 2 NADHs produced by oxidizing pyruvate to acetyl-CoA, and then 2 times 10 ATP for the acetyl-CoAs that get processed in the citric acid cycle and the electron transport chain. This totals to 30 ATP, which is significantly larger than the energy of glycolysis alone. In contrast to the 30 ATP we get from oxidative pathways, the anaerobic pathways that humans use, reducing pyruvate to lactate, produce much less energy. The NADH we would have gotten energy from in glycolysis needs to be oxidized by the pyruvate instead, which eliminates any energy gain from the NADH and any energy gain from the pyruvate. We're left with only the ATP produced directly from glucose, a measly 2 ATP. Anaerobic conditions are much worse at giving us energy. So why do we go through anaerobic pathways? If a tissue is temporarily depleted of oxygen, we want it to temporarily function without oxygen, even if it's far less efficient. This is especially important for muscle tissue, where activity can quickly use up all the available oxygen. If you couldn't process any energy anaerobically, your muscles would seize. Or, if your brain ran out of oxygen, you'd stop being able to function if you couldn't do anything anaerobically. We make lactic acid, but yeast and some bacteria, and even goldfish, can make ethanol instead of lactic acid by reducing pyruvate. The goal for the organism isn't the ethanol, even if it is a brewer's goal. The organism makes the ethanol to allow it to restore the NAD+ that it needs for glycolysis, and allows it to continue to survive in temporarily anaerobic conditions. Notice that the end result is the same for the lactic acid route and for the ethanol route. We only get the ATPs directly produced from glycolysis. We don't get energy from any other source. Again, 3 fates are possible for pyruvate depending on the organism and conditions, but oxidizing pyruvate to acetyl-CoA allows us to enter central metabolism and get much more energy than any of the anaerobic pathways.